very detail doing practical demonstration today. And each step is a full protocol. There are certain reagents, chemicals, buffers, which we have to add at any step and to get the sequences. So far, I think this is a very brief. If you have questions, we can discuss now.
Here we break samples by sonication or by
here we create design. For sonication, we have a sonicating machine. We need a sonicating machine. So enzymatic is an alternative way, which is less expensive and less laborious also. Very straightforward.
So you use general methodologies or what? How you
These analyses are provided by the manufacturer. Like Mina, they have their own, and they have their own. Oxford Nanopore, they have their own, and usually they do not disclose which specific enzyme is located.
The third is like the ligations you are going to make at the end of each fragment.
Exactly.
Is they provide a kit for that too.
Exactly.
And
so for all these modifications, the company provides a kit. We have to follow the kit protocol. Which we will do today, inshallah.
I have one question. How do we differentiate clusters on the slide on flow survey?
This one?
Yeah.
So this is basically it takes place in India. So these are the clusters.
Okay.
So when these clusters are generated, so you can just imagine on this glass chip, which is a flow cell, there are millions of clusters. Right?
Clusters according to the this is
No. So
the
number of clusters is dependent on the kit which we are using.
no. In Nano, we already added this is in the piece can go back.
In Nano, there are no clusters.
Okay.
So this is why we will. So this is a totally different technology. Minon is a totally different technology. In case of Minon, so this is a one. You can say this is a membrane receptor through which a DNA fragment can pass on, right? So, and this is the membrane, right? And there are thousands and hundreds of receptors like this. Okay. So on this membrane, which is present in the benign flow cell, there are thousands of receptors, right? So through each receptor, the DNA fragment passes on, and each receptor produces a DNA sequence, which is recorded in the FAST2 format. So this is the beauty of machine, that there are thousands of receptors here. So through each receptor, a different DNA fragment is passing on. So the machine is recording the sequence of each receptor simultaneously. So just you can imagine this is maybe this is a circle or something, some other shape. This one shape represents one receptor. So you can just count how many receptors are in one row and how many receptors are in one column. So maybe there are thousands of receptors, and through each receptor a different DNA molecule is passing on, because in laboratory preparation, as I have shown, yes, in laboratory preparation this genomic DNA has been transformed into thousands of fragments of different sequences. So these fragments one by one pass through the receptor which is present on the membrane. So there are thousands of receptors, thousands of different DNA molecules pass through the receptors, and through each receptor a different DNA sequence is generated, which is recorded in the FAST2 format, this one. So this DNA sequence was produced from one receptor. So when first base passed, the receptor detected that this is, I think, G. Yes, this is G. The receptor detected the first base is G. On the same fragment, the second base was A. On the same fragment, the next base was C, C, and so on. As the fragment was passing to the receptor was recording the base of fragment. So this sequence belongs to one fragment, right? For the second fragment, this is the sequence on second line.
But can you read the as NB501812?
This one,
This one is same for the second line, for the third
maybe
line.
there is some. No. You can see 15775.
So that's the difference between.
So this is the code for. So the machine assigns a unique code to one receptor, a unique code to next receptor, a unique code to all these receptors.
The receptors are chemical or what?
The receptors are just like membrane proteins. The cell membranes have a lot of proteins. So this is a specifically designed engineered protein receptor through which the DNA fragment can pass on, and there is a small variation in the voltage across the membrane. So the beauty of this receptor is that when a base passes through the receptor, the variation in voltage is different for all the four bases. If this variation is not different, how machine can differentiate whether this is A or C or G or T, which base has passed on? So based on the value of variation of which is produced due to each base, the machine records which base has passed on.
And how we do multiplexing different patients with the same.
So for multiplexing, I have just yes. For multiplexing, we use indexes. So as I told, the index has three portions. One which is complementary to the adapter, which we ligate in the first step, and then this is the index sequence, the middle one, and the last one, this is P5 or P7. So this middle sequence, which is index 1 and index 2, so this is different for different patients.
This one is the England.
So same is for. So difference is that the sequences are different, but in case of and torrent, instead of P7 or P5, there are, I think, adapted proteins which direct the fragments to the code channels. So based on these sequence of these indexes, the machine's software can separate different samples.